Good morning, everybody. We're going to work on Chapter 17, which is Transcription and Translation. So this video is just going to focus on the transcription part because with this program I'm limited to only 15 minutes, so I'll do the two separate. Okay, so just a reminder, we're going to skip all these review things in essence of time here. Okay, so just remember from regular biology we talked about how DNA and RNA are different, right? DNA is double-stranded, whereas RNA is single strand, which allows it to leave the nucleus. That's the important part of that. DNA has the sugar deoxyribose, whereas RNA has ribose. It has a U instead of a T, and then of course it can leave the nucleus, as we already said. Okay, so in regular biology, when we talked about transcription, we kind of just learned that it was turning DNA into RNA. It is actually much more complicated than that. So let me kind of get started with that. So in a prokaryotic cell, remember, which is like bacteria, it is basically what we learn in regular biology. You take the regular DNA strand, you create messenger RNA, then it heads over to the ribosome to make the protein that is needed immediately. But when in a eukaryotic cell, we actually have a couple more steps involved. So here's the transcription that we learned, but if you notice, it turns this into something called pre-mRNA, and then it kind of processes it, does a couple more things to it. It's going to put something on the tip and something on the back end, which I'll show you in just a second. Then it heads over to the ribosome for translation. So I kind of want you to think of this like we're making a film, right? And we filmed thousands and thousands and thousands of hours of movie material, and then you have to go through it and kind of cut out the scenes that you don't want. This is kind of like how I think of transcription in eukaryotic cells. Okay, so we've already gone over all that. I'm sure you're pretty familiar with that. And something that's new again, remember we can only add nucleotides to the 3' end. Okay, so that's the side that it's going to create the new messenger RNA from. But we don't have to worry about Okazaki fragments and all that because we're making RNA, not DNA. Okay, so here's the first picture of that there. And I want to show you this first. The template strand, remember that's going to be that 3' end. And there are actually 3 steps to transcription: initiation, elongation, and termination. So to make things really simple, initiation means you're going to start it. Elongation is when you're going to split open your DNA and start copying it. And then, of course, terminate means to end. Okay, so let's look at the first step there, initiation. Okay, this is something new, and it's actually kind of funny, and most kids memorize it because it sounds funny. We actually put a promoter on the messenger RNA that is called the Tata box. And the reason it is called the Tata box is because the first 4 letters of the DNA are T-A-T-A. Okay, so we're going to have an initiation complex, which includes that Tata box and a couple RNA polymerases, which are just some enzymes. So let me show you that in picture form. Okay, so here are your promoters. There's your Tata box. Okay, that with this initial strand here, and this is the starting point. So here is where you would actually have your AUG. Remember, AUG is the start codon, the methionine. Okay, so we're going to add different transcription factors here, and it's almost like so this thing knows where to attach. Okay, and notice here is that whole complex. So your Tata box would be way over here, and then here's your initiation complexes. Your messenger RNA doesn't actually start right here until you see that reddish pink thing there. Okay, so that's initiation. All right, then the second step is the elongation. This is where we're going to open up our DNA, and the DNA exposes about 10 to 20 base pairs at a time, because if it does too many, it could get messed up or mutated or damaged. And then in most eukaryotic cells, we can do 60 nucleotides a second. So it's actually a very fast process. Okay, so then termination, if you remember from our amino acids, we actually have 3 stop sequences. And these are the ones that are actually the termination thing. Okay, so what's going to be, so the Tata box is at the beginning of the messenger RNA strand. This one is going to have a poly A tail on the end of it. So what does poly A mean? It means there's going to be a bunch of A's, a bunch of adenines at the end. So let me show you that. Here's the poly A tail. Look how many there can be. 50 to 250 adenines at the end. Now, why would it do that? Well, a lot of scientists think, so like sometimes when you send the messenger RNA, like the last part of it actually gets damaged. So maybe that tail is on there. So if part of it gets cut off, it's not actually any genetic material. Okay, and then notice it also puts a cap on the front of it. Okay, it's guanine with a bunch of phosphate groups. This is called the 5' cap. So again, this yellow portion here and yellow portion here are not actually genetic material at all. It's almost like a protection on both ends. So this is actually the codon that we're going to, these are the codons that we're actually going to turn into a protein. Notice how small that is compared to the entire messenger RNA strand. Okay, and again, they think that's for protection purposes. Okay, so what we're going to do is we have to modify the RNA a little bit. So let me show you a couple new terms here. So remember, RNA splicing, it's just like what I said with the video. If we have a full-length movie with thousands and thousands of scenes and we only want to use part of them, we're going to have to splice some out and then cut and paste the rest of it together. Okay, so the messenger RNA itself is probably 8,000 nucleotides long, but the only part that actually makes a protein is about 1,200 nucleotides long. So you're talking, what, 6 to 7 times more information that is actually going to be coded. Okay, so let me show you those. And this is something that I always get mixed up still to this day, so I want to make sure I make it clear for you guys. So these introns, these things that are pink here, are non-coding segments. So that means they are not going to be expressed. They are not going to be turned into a protein. Okay, the exons are what is going to be actually expressed and leave the nucleus and turn into a protein. Now, how I get that mixed up is exon, I think of exit. Like these are the parts that are going to be cut off that we don't need, but it's actually the opposite. The introns are what is not needed. The exons is what is needed. So I tried to put that very simply here. Exons are what will be expressed. And this is what exits the nucleus. So the exons are what we need. The introns are what we don't need. Okay, so as you can see here, you've got your long pre-messenger RNA. And once all of this junk is cut out, you're left with the cap, the tail, and the actual gene that you want to create right here. Okay, so something else that actually cuts these introns out is called a spliceosome. I think it's pretty easy to remember because of its name, but just remember this is the one that cuts out the introns, leaves the exons, and then kind of like glues them together. Okay, so that's transcription in a nutshell. A lot of new material in there, but the basic concept is the same. Okay, so what I'd like for y'all to do is try and answer the free response question that I am putting up here with this video, and then go ahead and move on to the translation video, and then we'll be done with the basics of DNA, and we'll head into mutations and like modern DNA technology, and then we'll be done with what is fair game on the exam. So email me if you have any questions.